This is a structure of glomerulus and the Bowman's capsule. As I have already mentioned that the Bowman's capsule is made up of two layers. It is a double layer cup-like structure. The outer layer or the parietal layer is made up of simple squamous epithelium, and the inner membrane or the inner wall, known as the visceral wall, is made up of podocyte. The podocyte rests on foot-like process called the pedicel. In between the parietal layer, or we can say that in between the blood capillaries and the visceral layer of Bowman capsule is present a basement membrane. In this diagram, the parietal layer, which is made by simple squamous epithelium, can be seen as the rectangular outer blue colored cell. The visceral layer, which constitute of the podocyte, it is the specialized cell. It is shown by blue color also. The basement membrane is shown by the brown color, and the blood capillary is shown by the red color. Here is also the structure of glomerulus, showing also the proximal tubule and the distal convoluted tubule. The blood capillaries, the parietal layer of the Bowman capsule, the inner visceral layer showing the podocyte and the pedicel, the basement membrane, and the capillary known as the glomerulus capillary. The basement membrane is shown by the bluish, light bluish color. Parietal layer shown by again the large squamous cells, epithelial cells. The podocyte light green in color, having pedicel. The capillaries here shown with the light red color. So this is the structure of Bowman's capsule having the glomerulus. As we can see in the typical structure of a neuron, it comprises of a Malpighian body, and we know that Malpighian body comprises of the Bowman's capsule, which is a double-walled cup-like structure. It has two walls or two layers. The outer layer is called the parietal layer and the inner layer is called the visceral layer. The visceral layer is made by a special squamous cell called podocyte. This is the structure of the Bowman's capsule. As you can see, there are pores of glomerular endothelial cells which prevents the filtration of blood cell but allow the components of blood plasma to pass through it. Then it has the basal lamina of glomerulus. It prevents the filtration of larger proteins. Then there are the podocyte of visceral layer of glomerular capsule. So it is through these pores or filter fenestration that the blood is passed through and the larger particles gets filtrated. It is the section where all the three structure. First, the capillaries. Second, the filtration membrane or the basement membrane. And the Bowman's capsule or the cell present in the Bowman's capsule shown by the yellow coloration. The pink coloration is the capillary. Then the capillary can see the pores present in the capillary known as the filtration membrane. Then there is the basement membrane. Then is the Bowman's capsule and the specific cell present in the Bowman capsule. In the Bowman's capsule, you can see the foot process of the podocyte. We know that podocyte is the inner wall of the Bowman capsule. So there are foot process of the podocyte of glomerular capsule or Bowman capsule. Then there are the slit or the openings present in the pores present in the Bowman's capsule. The filtrate in the capsular space, that is, the blood passes through these pores. The second part of the Malpighian body is the glomerulus or the blood supply. As one can see in this picture, the efferent arterioles having the blood flowing through them. Through the Bowman's capsule, the filtration is happening. The Bowman capsule has got the pores through which only the plasma can pass. The red blood cells do not pass through these pores or the opening. Then there are finer pores through which only the finer molecules can pass and the proteins cannot pass. Here is also the diagram showing the structure of a nephron, the glomerulus having the capillaries or the Bowman capsule in which the capillaries are there. See the fenestration or the pores of the glomerular endothelial cell, which prevents the filtration of blood cell but allows the component of blood plasma to pass through. The basement lamina or the basal lamina of glomerulus prevents the filtration of large proteins into it. This is a schematic diagram showing the capillaries and the Bowman capsule, the fenestration or the pores, the foot process of podocyte, the filtration slit, glomerulus. We know that is the rounded tuft of blood capillaries found in the cup of Bowman's capsule. The renal artery is a branch of abdominal aorta. It enters through the hilus or the hilum divides and redivides into many branches. As I have already mentioned earlier, the capillaries reunite to form the efferent renal arteriole. It leaves the glomerulus. The structure and the expanse of capillaries I have already discussed. The proximal tubule or the renal tubule is made up of, we know, the proximal convoluted tubule, the Henle's loop, the distal convoluted tubule. It is a long convoluted structure, convoluted means coiled, that emerges from the glomerulus and can be divided into three parts: the proximal convoluted tubule, the Henle's loop, and the distal convoluted tube. The proximal convoluted tube, due to its proximity, that is closeness to glomerulus, it stays in the renal cortex. The second part, called the Henle's loop, because it forms a loop and was discovered by researcher Henle, it is known as the Henle's loop. It goes through the renal medulla. The third part of renal tube is called the distal convoluted tube, and this part is also restricted to the renal cortex, as is evident in this diagram. One can very easily see where these tubules are situated: the cortex and the medulla. In the medullary region, one can see the Henle's loop, the proximal convoluted tubule and the distal convoluted tubule are present in the cortex of the kidney. The distal convoluted tube is the last part of the nephron, and it connects and empties its content into the collecting duct that lines the medullary pyramid, as can be seen in this diagram. The proximal convoluted tubule is a highly coiled structure. It is lined by cuboidal cells. They are rich in mitochondria and have microvilli or brush border to increase the surface area and thus help in the active reabsorption. The Henle's tube descends into the medulla. The thin walled descending limb takes a hairpin turn, a U-shaped loop, and ascend back towards the cortex. The ascending limb is thick walled. The ascending limb is not permeable to water. The Henle's loop mainly meant to reabsorb water or concentrate the urine. Convoluted tubule leads to straight, large collecting tubule. Many nephrons open into the collecting tubule. Many collecting tubules join to form a collecting duct, which descends into the medullary pyramid. The medullary pyramid are basically bunch of these collecting duct, hence they appear to be striated. Striated. Striated means appearance of linear or line-like structure. The medullary pyramids are basically bunch of these collecting duct, 7 to 8 collecting duct draining from different nephrons join to form duct of Bellini, each of which opens into a minor calyx at the apex of medullary pyramid. This I have already mentioned. The efferent renal arteriole, which leaves the Bowman's capsule, break to form peritubular capillaries around the whole renal tubule. The capillaries around the loop of Henle are called the vasa rectae. Capillaries reunite to form a renal venule and carries the blood away from the nephrons. The renal venule join to form the renal vein, which passes out of the kidney to join the inferior vena cava. The renal tubule helps in the selective resorption of essential substances like water, glucose, and salt. There are two classes of nephron, as I have already mentioned, one present in the cortical region and the other present in the junction between the cortex and the medulla, known as the juxtamedular nephron. Physiology of excretion or the ultrafiltration. This occurs in the Malpighian body or the Bowman's capsule and the glomerulus. The physical process where the blood gets rid of its nitrogenous waste or other waste is known as the excretory process, and this occurs by the process of ultrafiltration. The diameter of efferent arteriole, as is evident from the diagram, is bigger than the efferent arteriole, thus creating the pressure in the glomerulus. The glomerular hydrostatic pressure is the blood pressure in the glomerular capillaries, which is about 55 millimeters of mercury. The colloidal osmotic pressure of blood, which is 30 millimeter of mercury due to the presence of plasma protein. It opposes the glomerular hydrostatic pressure. The capsular hydrostatic pressure of glomerular capsule is caused by filtrate that reach into the Bowman's capsule, and it is about 15 millimeter of mercury as opposed to the glomerular hydrostatic pressure. So we can say the net or the effective filtration pressure is the glomerular hydrostatic pressure minus the Bowman's capsule osmotic pressure plus the capsular hydrostatic pressure.